Ah! There. Okay. Sorry, guys. I got wrapped up. Okay. Lecture 9. I hope not. Okay. I just forgot to change the title slide. Okay, guys. So this is the last lecture before the exam, and whatever we finish today is what will be on the exam. So if we don't get through it all, don't worry about it. Okay, so this is the list of what you should know. People say things to me like, please tell me this is right. Yep. People say things to me like, oh, what's in the exam? Just keep looking at them. Yes, it looks like a lot, but it's only like one main process that we're talking about. Kind of. Okay. Business. Exams on Monday. Materials through today. Send your multiple choice questions by the end of the day Friday. Multiple choice questions. Remember I said that was an option? Maybe some of you don't. If you want extra credit, another opportunity is to write your own. You're looking confused. Nina's confused? Yeah. Okay. So we're starting to know people's names. It's so exciting. Yeah, one way to earn extra credit is to write me a multiple choice question. And if I use it and it's perfect, you get 2 points, plus the points that you get on the exam for knowing the answer to your question. And then 1 point if it's pretty good, but I need to do a fair amount of fixing. Now, in the past, students have been smart and shared all, like made this big spreadsheet full of all the multiple choice questions, shared it in hopes of having a copy of the exam before the exam. That doesn't work very well because it's really hard to write a good multiple choice question. And sometimes the people writing them don't know what they're talking about. And so then you're getting just flooded with all this garbage. So you can do that if you want, but I just warn you to spend your time studying rather than trying to find a way to cheat. I'm not saying you guys would. This was just last 2 years ago. One sad moment. Okay, so that's how it goes. So you have until Friday. End of day. And that's because I need to print it before I give you the exam. Okay? Okay, so we finished it with the secretary pathway. We know everything we need to know about that, right? And so we started on Monday, talking about transport of proteins to other compartments, specifically the nucleus, and now we're going to also talk about ER. So this is where we ended, right? So there are different ways of moving between compartments. There's vesicular transport, which is the secretory pathway. Gated transport, which is there's actually something that's preventing movement of the protein from one side to the other, such as cytosol to nucleus, and we'll talk about in detail shortly. There's transmembrane transport, which utilizes protein translocators to pass the protein across the membrane. And that would be what? What did we already learn that uses transmembrane transport? Into the ER, right. And then we're going to learn about the mitochondria today, hopefully. And then then so that leaves me with this question of what determines the correct address for any given protein? Some kind of signal sequence. And actually, I should have put this slide in the beginning, but I put it in the end. Let's see. Let's go. It's all the way to the end. Oh my god. Look at all that. Yay! That. So in the book, Table 12.3, they have a nice chart that tells you all the different sequences and how they function. And so there you go. Don't memorize this, but just understand that there are different signal sequences. Now, we had this conversation today in my office, and I think of signal sequence as going to the ER specifically. But actually, I looked it up after we spoke, and the textbook does refer to a signal sequence as any sequence within the protein that directs it somewhere. But there are different kinds of signal sequences that might have different names, and we'll talk about that shortly, and because we're going to talk about nuclear localization signals, which is a kind of signal sequence. So let me go back. So those are all the different types of signal sequences, and you've already learned about a bunch. Okay, so this is the nucleus, right? And we talked about this on Monday, so we're not going to repeat it, about the type, why the proteins go in and out of the nucleus. And it's because there are proteins that need to work there, like polymerase and stuff like that. Why do things come out? Remember, RNAs need to come out, and they're bound by proteins and all sorts of things. And we talked about how proteins get in and out of the nucleus. They enter and exit through nuclear pore complexes, one of you. And that the numbers of nuclear pore complexes change dependent upon whether there's a lot of in and out traffic going on. And so this is the nuclear pore complex, and it has these fibrils which have these FG repeats, and that'll come up again soon. So this is this gated transport. This is an active transport of proteins across through the nuclear pore. So they're not going across a membrane, not going through a membrane. There's an actual pore that they're going through. And only very small molecules can diffuse through the nuclear pore, things that are 5 kilodaltons or 9 nanometers or less. And just for example, DNA and RNA polymerase subunits tend to be much, much larger, on the order of 100 to 200 kilodaltons or so. Ribosomes are fairly large too. And so things need to be transported in and out. And then we spoke about why you would want to regulate what is going in and out of the nucleus. And that's because you can control things. Like, so for example, gene expression is regulated by access of transcription factors to promoters. So one way to keep gene expression off is to keep that transcription factor out of the nucleus, right? So we spoke about that. I'm kind of flying through because you all heard this before. And then this is nuclear localization signal, lysine and arginine rich, right? So this is a T antigen in the nucleus, and when you mutate even just one amino acid, here they've mutated a lysine to a threonine, you no longer get nuclear import and your cytoplasm. And this is showing how gold spheres that are covered with an NLS will make their way through nuclear pores. Okay, so this is where we stopped. Everyone feeling oriented, like they know what we're talking about? Sure. Okay, so how do nuclear proteins get in and out? Well, they're usually associating with either importance or exportance. So we'll start with talking about import. Nuclear import receptors, or importance, I might use those words interchangeably. Nuclear import receptor or important, same thing. They bind to the NLS in the cargo protein, and they also bind to these FG repeats. on the nucleoporins, and that's how proteins get brought in. So the protein doesn't come in by itself, but it needs to interact with an importin or a nuclear import receptor. And so what this protein's showing is just that, so in all of these cases, the S's here are the importans, and then you have got your cargo protein. And it's just showing there's different importans that bind to slightly different NLSs, but either way, nuclear import receptors can get into the nucleus. So as long as the protein can bind to it, you're good. In some cases, the cargo protein won't have an NLS, but it will interact with a protein that will have an NLS, and so this is the nuclear import adapter protein. And so there's a third protein involved. Okay? Anyone have any questions about this? Yes?
So technically, for that next one, if it doesn't have anything for it to bind to is something that's not supposed to go into the nucleus able to bind to one of those that can break it back one of those something and do it something?
Okay, so the question is, tell me if I'm getting it. So you're asking if things can get into the nucleus that shouldn't get into the nucleus just because they can bind to this adaptive protein. Yes, and viruses like to do that. Yeah, sure, but like ideally only things that should bind will bind, but there are viral proteins that have the ability to interact with these kind of adapter proteins and get into the nucleus or out of the nucleus depending on what they need based on that mechanism. Yes, that's a good question. So these nuclear import receptors are going to facilitate the nuclear import of a cargo protein and then return back out to the cytoplasm. Right? So they're like little shuttle bus. Okay, so I'm going to go through the mechanism on the next few slides, and I'm going to keep checking in with you because sometimes this can get confusing. Sometimes it's really simple. Sometimes it's confusing. So let me know how it's going. The basics, though, is just you've got this protein and has the NLS, and it binds to an importin, and then it interacts with these FG repeats in the nuclear pore, and then it goes in to the nucleus. Simple, right? And then there's RAN, and RAN is involved with release of the protein from the importin inside the nucleus. Okay, that's the gist of it. Yes.
What are the
These are on these. So remember before I was showing you this picture, and I said these are your nuclear pores, and there are these fibrils that kind of stick off, and so these contain these FG repeats, and since the importins can interact with them, that's kind of how they walk through. They kind of hop through the nuclear pore on these FG repeats. Yeah.
Do the F and the G correspond to amino acids?
The F and the G, phenylalanine and glycine. Yeah, they correspond to amino acids. Yeah. Any other questions? Okay. So that's the gist of it, but it's of course a little bit more than that. So RAN is another one of these GTP binding proteins. What's the GTP binding protein we just talked about the other day? RAB, right, okay. So if I say it's a GTP binding protein, you should think it's a molecular switch and has some conformational change associated with whether it's GTP bound or GGP bound that affects whether a function is happening or not. With RAB, it was about targeting vesicles to membranes. With RAN, it's about release of cargo from importins. Or not even release, but it's about regulating the interaction of importins or exportins with the cargo. So in this figure, it's showing your protein with your NLS, and it's binding to your importin. Here it's showing alpha beta. It's a dimer. That's okay, right? it goes in through the nuclear pore. Once it's inside, you have RAN, which is GTP bound, so it's on, it's active, and it binds to the important, which allows for release from the it releases the cargo in the nucleus, and then RAN goes back out into the cytoplasm, bound to the important, right? And you want your important to go back out, right? Right? Maybe? Yes, no, maybe? Right? Because if they all end up in the nucleus, that's not good. Because how could they import more proteins if they all end up in the nucleus? Okay, so RAN ferries the importance back out to the cytoplasm. Okay? How are we doing? Yeah, maybe. Okay. I got some thumbs up. Okay, so RAN gap, so whenever there's a GTP binding protein, there needs to be a gap or something like that, right, to regulate its activity. RAN gap stimulates GTP hydrolysis, and RAN gap kicks off the GDP and restores the GTP in the nucleus. So RAN gap is associated with the chromatin, so it's always in the nucleus. It's not going in and out. And so you always have this active RAN in the nucleus ready to go. RAN gap is out in the cytosol all the time. It doesn't move in and out. So it's always hydrolyzing the GTP. Okay. So let's talk for a minute. Yes.
So does it give Jeff?
Yeah.
Does that help in releasing the protein?
So RAN gap keeps RAN GTP active so that it can, so let's go back a step. So it can bind, so remember what RAN does when it's GTP bound, it binds to the important and that facilitates release. So indirectly, yes, RAN gap is what keeps RAN GTP bound, and that's what allows it to bind to the important and release the cargo. Okay? So it's just, there's two players, there's two main players here, RAN, and then the RAN gap and the RAN gap. Okay? And so the RAN gap and the RAN gap are going to determine the activity of RAN. Any questions? Yes?
Yeah, which one's the nucleus again?
RAN gap. It's actually bound to the chromatin, so that's how you can remember it. Because chromatin is in the nucleus, right? Any other questions? Okay, so now that was nuclear import. Let's talk for a minute about nuclear export. So, yes, gap is always in the cytosol, gap is always in the nucleus. So nuclear export is similar. You have a nuclear export signal, just like there's a nuclear localization signal that determines whether or not you can interact with an important. There's a nuclear export signal that determines whether or not you can interact with an exportant. It's so distracting when people walk out like that. What? Did I say something? Okay. I just want everyone to be happy, okay? No one to leave. No one's allowed to leave. There she is. I thought you left. Okay.
I just need Sorry.
Okay. So with nuclear export, sorry guys. With nuclear export, you've got your protein that's in the nucleus, right? You're exporting it out of the nucleus and needs to get out. So here's your protein right here with your nuclear export signal, and it needs to bind to your exportin. And in this case, RAN GTP allows it to bind. Okay? So it's not inducing the release, but it's actually encouraging the interaction between the protein with the nuclear export signal and the exportin, and brings them out of the cell. Right? So there goes RAN GTP out of the cell with the protein that needs to be exported and the exportin. And then out here, you have your gap, which hydrolyzes the GTP, which then releases it. Okay? So here you have GDP, RAN GDP, and you have your exported protein release, and then your exportin can go back in to the nucleus because you don't want all your exportins outside of the nucleus, otherwise they won't be able to export anything. Okay? How are we doing? Yes?
What did you say ran gap hydro?
So gaps and gaps. Gaps and gaps. Gaps. So gaps hydrolyze GTP and gaps exchange GDP for GTP. Yeah. And so whether it's RAB or RAN or any other GTP binding protein, and it'll come up over and over again through the semester, always keep that in your head that it's going to work the same. Okay? Yes?
So is the exporting going to go back into the nucleus?
Exportin has to go back into the nucleus. Yes. Yes, because you always want to keep your exportins in the nucleus so they can bind to things with nuclear export signals and get them out, and you want to have your importins in the cytosol so they can bring cargo in.
So how does it get back in?
It has its own nuclear localization signal. Yeah. Question is, how does the export get back in? It has its own nuclear localization signal and interacts with its own import. Yeah. Yes.
So how does like the red gap tell? Does it tell like if it needs to do something like if it's finding the export term? Like,
Well, think about where things are, right? RAN gap is always on the outside, so whenever, RAN gap is on the outside. Whenever it encounters RAN, GTP, it hydrolyzes. Okay? And so if RAN GTP is bound to an exportin, that will result in dissociation. And if it's bound to, same thing, right? If it's bound to an importin, that's when the importin's coming out with RAN GTP. And then it's dissociating the importin and the RAN GTP. Or the RAN GTP. Okay? So RAN gap and RAN gap live where they do and always are going to do what they're going to do. So RAN gap is always going to hydrolyze the GTP on the outside, and RAN gap is always going to exchange it on the inside. Yes?
Is the GDP synthesized in the cytoplasm and brought to the nucleus as GTP or GTP?
So where is GTP made and GDP? Is that your question?
Yeah, so the GDP, is it
Oh,
synthesized
God.
from the GTP?
Wait. So come again?
So is GDP synthesized anyway to from GTP?
So
Does it go back into the nucleus and then?
Okay, so GDP becomes GTP when another phosphate group is added. So are you wondering how the GDP and GTP is what it is?
There are just like two separate processes that were occurring and just one.
Is it the GTP you're wondering about or the RAN GTP? Okay, so that's a different, right? Because GTP is just a nucleotide that's binding to RAN. So, okay. So your question is the RAN GDP. So the RAN GDP is always going to be on the outside, right? No. Wait, I don't understand your question.
Never mind.
Okay, do you want to come see me later and we can discuss and maybe you can clarify? Or did anyone around her quick understand? No. Sometimes the neighbor understands. It's like, no, what she means is. Okay, we'll get to it. We'll hug it out. Okay. Any other questions? Yes.
if the exoporn has NLS, does the nucleoporn have an NES?
If the exportin, so if something's going to make it into the nucleus, then it must have an NLS, and if something's going to get out, then it must have an NES.
So is it the protein that binds to it that makes it go into it?
So
In
anything,
and out?
yeah. So they're going to also bind to importins and exportins to get it out. They have their own. So it's not like there's one importin. So the question is, does the importin is in, and it needs to go back out? Does it have an NES? Right. That's your question. And does it bind to a nuclear export receptor, right? That's your question. And so yes, and all of these bind to import receptors or export receptors to get to where they need to go. And it's not just that there's one, right? There's many importans and many exportins that bind to their own classes of proteins and import and export them. But they recognize them by binding to their NLS or NS. Make sense? Okay. Woo! Anything else? Yes. The plain GDP? I'm guessing that's confusing, right? So well, the GDP has to be made into GTP by adding a phosphate group. And so then, I think it's throughout the cell and it's everywhere because it's tiny, it's a nucleotide, right? Yeah. Any other questions? It's like oral exams every day. Okay. You still have questions?
No. We were talking about the name and it's all like the same.
I know. You just have to remember import and export. I know. Why does it have to be ran, gap, and ran, gap, and ran, and ran, and ran? Right. I know. People always confuse that. So if you were to get your functions down, then you should be good. But people always confuse it. Okay, we did that. We did that. Right. So nuclear export. Okay. So here's the picture with everything on one page. And so what I was telling someone earlier today is please don't limit yourself to Albert's here. A lot of these figures were taken from the cell and molecular approach, which is available free online through NCBI books. I like the way they explain it better than Albert's. And so when I read through it in Albert's, I was like, okay. But then I wanted to understand it more deeply and have better figures. And so I went to this book. So please go to the multiple books that are available and really try to understand it. Okay? Yes?
I have a question. I think I have a question. All right, on this picture, you see it has GTP and then it goes out and it goes into GDP. What causes that?
Ran gap.
So that adds phosphate group.
No. GTP to GDP, that's hydrolysis.
Right.
And so it's removing the phosphate group.
Okay. So what's adding it in nucleus?
There's a gap in here. So that goes back to this one. See the RAN gap? That lives here on the chromosomes. And the RAN gap lives out here. And so RAN gap is always always hydrolyzing GTP to make it into GDP. And RAN gap is always kicking off GDP and replacing it with GTP.
So basically, in the inside, it's just adding a phosphate, on the outside, it's taking it. Well,
It's not adding a phosphate. It's kicking it off and replacing it with GTP.
okay.
Yeah. Okay. Huh? It does not. RAN gaps do not add phosphate groups. They remove GDP. They kick off the whole molecule and replace it with a GTP. That sounds like the same thing, but it's different.
You need to worry about the excess GDP.
Huh?
When it comes in, if it just takes the GDP off and put it in GTP, that means it's like excess GDP. You need to worry about that.
So you worried that there's all this GDP floating around here that's gonna make trouble? Okay, so the question I got is, do we need to worry about this GDP making trouble? My guess must be that the affinity of GTP for RAN GTP must be higher than GDP, but I don't know. Because are you worried that there's so much GDP that maybe it'll kick off the GTP that just got put on?
Yeah, because if it hydrolyzes it outside to GDP from the GTP and then the inside just takes it off and replaces it. So when it takes it off, do it go back out?
I'm guessing there's whatever enzyme that's responsible for making GDP into GTP, there must be a lot of it, so it must constantly be converted back to GTP. Yeah, but I don't know the exact answer. That's my assumption. Okay, you?
Yeah. Okay. So what's the point of taking off a whole molecule when you just add a phosphate group?
So the question is, what's the point of taking out the whole molecule when you can just add a phosphate group? I don't know. I don't know. It must have to do with the enzymes, right? I mean, it must just have to do with the enzymes that are there, because what you're trying to do, there's specificity here. Think about it, let's think about it for a minute, okay? There's specificity here with RAN GAF for RAN, right? But there's less specificity for adding a phosphate group to things, right? So if it's just gonna bind to GDP and add a phosphate group, that's less specific and there's a lower level of regulation. So by having specific gaps and gaps for each GTP binding protein, you could specifically regulate that. Now you might say, well, why not have an enzyme that binds specifically to RAN and adds a phosphate group?
You're right.
I have no idea. You? Venus?
Oh, that was my question.
Okay. And I saw someone over here. Okay.
What do you mean by
What do I mean by restores GTP? It means that this is gonna come in RAN GDP, and then the GEF is gonna restore it to GTP form by kicking off GTP and adding GTP. Pretty much because you've got a gap out there, so it's gonna wanna hydrolyze. Yeah. Okay, yes.
So is RAN only the bind to export or prevent its GTP now?
So that's specific to the process, right? Oops. Right, so in this case, RAN is gonna bind to the important inside the cell when it's GTP bound, and once you have GTP hydrolyzed, it releases it. And in this case, RAN is gonna bind to it's actually gonna bind to the cargo plus the important and only the GTP bound state, and when it's hydrolyzed, it's gonna release it. I guess it's slightly different because in one case, the cargo is being released because of the binding, because it's switching, it's like swapping out the interaction. And in the other case, it's actually encouraging an interaction and picking up a cargo. So I don't think you can completely generalize it. Okay, so let's go here. This is a picture of everything. This always generates a lot of discussion. Okay. So you can think of it as RAN GTP is regulating the transport of nuclear or cytoplasmic cargo, and it's providing directionality. So with nuclear import, let's see, where do we want to start? With nuclear import, you have your protein with your NLS that binds to your important, and then it comes in here, and then RAN GTP is going to bind, and that's going to kick off the cargo, and then it's going to take out, then the important's going to get taken out into the cytosol, and then you'll have GTP hydrolysis and release of the nuclear import receptor. And then with nuclear export, you're going to have your RAN GTP, but your RAN GTP is going to encourage complex formation between your export and your cargo. So all three bind together and make this complex, and then come out, and then you're going to have GTP hydrolysis, which allows for the release on the outside. Okay. Are we good? And the fact that you have your RAN GEF only on the inside, that means that you're only going to have binding of your RAN GTP to your important on the inside, allowing release, right? Because you need your GTP bound state, and then because you have your RAN gap on the outside, you're only going to have release of your cargo from your export on the outside, because it's that GTP hydrolysis that's required for the release of your cargo on the outside. Questions? That's a wonder. Yes.
So RAN GTP always takes something out of the nucleus. It just, it'll vary on exactly what.
RAN GTP always take. Oh, I like that. I like that. RAN GTP always takes something out of the nucleus. Yeah, because here it's taking the important now. Well,
Yeah.
it's going out with the important. It's not necessarily taking it out because it doesn't have an export. Whatever. It's going out here, and then here it is taking out the exported material. I like that. I like that. Yeah. Okay, any other questions, thoughts, comments? Okay, let's keep moving. Okay, so, binding of the import receptor by RAND GTP alters NLS recognition site, which is what forces the release of the cargo. And so this makes a lot of sense when you stop and you look at it for a minute. So this will be your protein, right? And this will be your RAND GTP, which I guess is also a protein. But this is your cargo protein. And so this red loop here is the portion that wants to interact with. This is the portion that wants to interact with your import. So actually, let's stay away from here. Let's look over here. So here's your cargo protein, and here's your important receptor. And so you have your NLS bound to some part of your important, and it once. How do I get them turned around here? See, it's confusing for me too. Cargo important receptor. Once RAND GTP binds here, so compare here to here. See this red loop? That's your interaction. But once RAND GTP binds, that red loop, there's a conformational change that causes that red loop to switch over and want to interact with RAND GTP, and that's how your cargo is released. So it's this conformational change that's induced by RAND GTP binding. Okay? So you see that? Here's your import receptor, and all it is it's making your import receptor not able to bind to your cargo anymore and want to bind to your RAND GTP. Okay? And that's how cargo is released. Okay, any questions about this? Okay. Okay. So we've been talking about nuclear import and the importance of the NLS. So here, this is just an example. It's an example of how nuclear localization can be regulated by controlling the visibility of an NLS to the nuclear import receptor. If you can't see the nuclear localization signal, the important can't bind to it, you can't bring the cargo into the nucleus. And so this is in T cells, this example, where you have this transcription factor. And so normally its NLS is phosphorylated. And so that's why it's in this cytosol. But when you have an activated T cell, it activates this phosphatase. Don't worry about what it's called. And that removes your phosphate groups and exposes your nuclear localization signal. And so then it can bind to the important and be imported into the nucleus to activate gene expression. And so that's one mechanism that is used frequently, actually, to regulate gene expression. You keep your transcription factor in the cytosol until it needs to go into the nucleus, and you can keep it in the cytosol by phosphorylating and covering up your NLS. Okay? Does that make sense to people? And so here, there's a kinase in the nucleus, and wait a minute. There's a kinase in the nucleus that will phosphorylate. So then this phosphatase actually binds over here, which covers up your nuclear export receptor. And in the nucleus, when you have a resting T cell, so you no longer need gene expression, your kinase will become activated and phosphorylate your nuclear localization signal, and then your phosphatase, which was bound and obscuring your nuclear export signal, it'll be released, and then you'll get export of your transcription factor out of the nucleus when you need to turn off gene expression. Okay, so that's kind of a cool little mechanism. And it all revolves around whether or not your transcription factor is in the nucleus and whether or not your NLS or your NES is exposed. So you guys are looking like you need a break. Okay, so we'll have a break with a practice problem. So you're studying a viral protein because that's all I know about. You're studying a viral protein that appears to be facilitating the import of certain proteins into the nucleus. Okay, this is actually kind of what you were talking about. What mechanism might the virus be using to import these proteins into the nucleus? Design an experiment to test your hypothesis. That's part one. Another viral protein appears to be preventing nuclear export of a protein. What could be going on here? How would you test your hypothesis? And so just think about these concepts that we just talked about how importance need to bind to NLSs and exportins need to bind to nuclear export signals. And now so phosphatases, kinases can phosphorylate things and cover them up, and phosphatases can remove phosphate groups and all that good stuff. Okay, so take a minute to talk to your friends to do that, and then we will reconvene.
Okay, so it says, so you see that the virus is a flashing so it might be not So you're saying that the virus is, you can't essentially, so there's two things that are bodies from the brain get antibody or it can move to the essentially you can say the same thing about export. So well then again, if you're banning export, then you can something like how to inhibit rat yeah. Yeah, so if it's for the other one, it's also situation, mutation occurs, it's gonna change the shape of the function.
One circle around. Okay, guys. Okay. Okay, that was a really long break. All I did was walk around once. Okay, so I can hear that there's a lot of confusion, and that's why I try to go through this slowly. Okay, so you're studying a viral protein that appears to be facilitating the import of certain proteins into the nucleus. What mechanism might the virus be using to import these proteins into the nucleus and design an experiment to test your hypothesis? Now, this can go, you can go 50 directions. There's not a right or wrong, there's just, well, there could be a wrong. But as long as it's logical, then you're good. Okay, so what do you think the virus might be doing to import proteins into the nucleus? Be it its own proteins or whatever. Yeah.
And I'll assume.
So the virus is adding an NOS to a protein? How? Okay. So you're almost there, but I want more. Yes.
Is it making something, it's like making its own important, but it has a higher affinity for the cargo for the red GTP.
So you're saying maybe the viral protein is acting as an important?
And it's better
It's a better important? Okay, that is logical. Okay, so you're saying adding an NOS, you're saying acting as an important, yeah. I'll get you back in the, yeah.
Viral protein might be making adaptive proteins that bind to the nuclear import
So maybe the viral protein is an adaptive protein that's grabbing a protein and binding to the important? Yeah, that's logical. What were you gonna say?
Like, can it, I don't know, like mutate,
This is a protein.
important,
What else? You guys had something? Somehow increase activity of a kinase. A kinase that would be doing what? Okay, so I'm hearing increasing the activity of a phosphatase, but what kind of phosphatase? What's the phosphatase doing? Okay, so it's masking the exporting. I see where you're trying to go. Yes.
So the phosphatase might be removing the phosphate groups that are inhibiting, like blocking the nuclear relations.
Okay, so she's saying maybe your phosphatase is removing the phosphate groups that is blocking the nuclear import signal. Is that what you were thinking? Yep.
Maybe somehow like it's making like the lysine and arginine rich like sequence for the nuclear localization sequence, so it can go through the nucleus that way.
So that's kind of what you were saying, is it's providing an NLS, maybe?
Yeah.
So, okay. So what I'm hearing is, yep.
Well, you were talking about that one side, you were saying something with the S's, with the cargo, that it would be possible for them to like mimic and latch on to be important, so.
Right, so like an adapter protein.
Right.
Acting as an adapter protein. So these are all logical hypotheses and things that if you were sitting there in your lab talking to your boss about what your next experiment would be, you'd be brainstorming like this. But then comes the hard part is how do you test it? And so you guys are not bench scientists, you don't know all the methods, but does anyone have any thoughts about how they might, okay, so let's go with yours, you're providing an NLS, okay? So if you have an NLS and you think that is, you have a viral protein with an NLS in it, and you think that's responsible for protein X going to the nucleus, how would you test that? Yes?
Gel electrophoresis, take the virus by itself and run it through an agar and tag it with an NLS and see where it falls.
So you're saying, see if adding an NLS will change its molecular weight. But does that affect whether or not it's going in the nucleus?
Well, okay, so you take your control ginger virus by itself and then take it from the nucleus and then take it with an NLS and then keep it.
Okay, so you're saying fractionate the cell and add an NLS and see where the proteins go. Right? Basically. I want something else. Yes.
I was gonna say or mutate the lysine and arginine sequence and tag the DNA or protein and see where it goes. If it doesn't go into the nucleus, then tell that it was going into the nucleus.
Okay, so mutate the NLS in the viral protein that you think is doing this. Tag it with some kind of fluorescence and do microscopy, maybe, and see if it still goes into the nucleus. That's a good one. What else? I saw another one. Someone else? So what you were saying, you were very close. What you can do is, don't worry about, you can do the same thing, merging your method with hers. Mutate the NLS, and then separate the cell. You can actually do this into cytoplasmic and nuclear fractions. Run it on a gel and see where it is, because you can look for your protein. Yeah. Okay, good. So another viral protein appears to be preventing nuclear export of a protein. What do you think was going on here? Did you guys get to this one? Yeah.
Could the virus maybe in some way be affecting RAN gap? So like the cargo could be.
Oh, okay.
I don't know how we get in there and stuff, but could it possibly block RAN gap for us?
So is the viral protein affecting the function of RAN-GAF? Now, if the viral protein is affecting the function of RAN-GAF, which is possible, would it affect all export? So export of all proteins or export of just specific proteins? All. It would affect all because RAN-GAF is like such a huge regulator of export. So if you saw no protein ever coming out of the nucleus, then that would be a very logical assumption. So I saw a bunch of hands going up. Yes.
What if it's just affecting exporting and binding
What if it's affecting export and binding to the RAN GTP protein complex?
yeah, because it can't be.
Yeah, that's very logical. Maybe. Maybe what else? Yes. Maybe the viral protein is positive protein to misfold. So what would that say? No. I mean, that would probably result in being degraded at some point long before it ever gets there. But that's if the protein went away. And the protein was completely gone. But here, it's not getting out of the nucleus. So it's still stable, it's just stuck in the nucleus. Yes?
Maybe it could have a post-translational modifications that would allow it to not bind to the export protein, so it would just stay inside the nucleus and not get out.
So maybe it's kind of like what they were saying is increasing some sort of kinase activity or something that will block the nuclear export signal so it can't get out. Okay? Yes?
I still have a question. So does exported work the same as imported, where it's different, they can bind to different export signals
Yeah, so they're nuclear export signals, and that's where the,
and the different exports?
yeah. Yeah, yes.
What if it mutates or affects the export in itself or the NFL signal inside of the export, so that it doesn't even go to the nucleus to take out the protein it's supposed to?
Okay, so you're going way far back, 10 steps back, where viral protein is somehow affecting exporting thing, so there's not even any exporters available to export proteins out.
Well, do exporting take any protein out, or is each export specific to different?
There are different, so the question is, do exportants take any protein out, or does every protein have its own export? There are different classes of exportants that recognize different nuclear export signals. So you would say one per protein, but maybe a whole class of proteins would be in there. Yes.
You always say that the cell likes to conserve energy, right? Is there a way that the virus can bind to an export without having to use the energy of hydrolysis with GTP and GDP and then get out that way because it's using less energy and therefore the other proteins are staying in and that's able to account?
So the viral protein is taking the job of RAN GTP. Is that what you're saying?
Yeah.
It's acting like RAN GTP, but it's not RAN GTP. Why not? I mean, you would have to do some kind of experiment to test that. That seems like a really hard experiment to do off the top of my head because you have to control for so many things that are involved in it. But it's not out. It's not impossible. Okay, some more people. Yeah.
Is there any way that we can affect the nuclear pore itself with the receptors when we put it pore?
Is there a way that we can affect the nuclear pore itself so that nothing can go in or out? That sounds like a drug thing, right? Like you use a drug that prevents. I have no idea. Yeah, where are you going with it? This idea, there's no turning.
Yeah, it's like late in the afternoon and it's like, just thinking, tossing it out.
Okay, so in the interest of time, I'm going to say, oh, one more.
You just cleave the nuclear exports.
So the virus the virus somehow removes the nuclear export signal? Maybe. The thing about nuclear import and export signals is they're not always at the beginning or end, right at the beginning or end. Sometimes they're not in the middle, but not right at the beginning or end. Okay, in the interest of time, I'm going to move forward. Okay, let's talk quickly about the mitochondria. Okay, so that was the nucleus. Okay, there are also proteins that need to work in the mitochondria, and we'll see how far we get in 5 minutes. You guys did this all on purpose. You didn't have to study to it. You're like, let's ask lots of questions and be really awesome, and we won't take it. So let's just talk briefly. Import into the mitochondria, right? So you have your mitochondria, and proteins could function as membrane proteins in the outer membrane, as membrane proteins in the inner membrane, in the intermembrane space, in the matrix, right? So there's a lot of places that you can end up in if you're a protein, depending on your job. So different proteins reside in the different compartments and need to get to the right place. And so how does that happen, right? So there are key players: signal sequence, translocators, chaperones, and energy in the form of ATP hydrolysis and a proton gradient. Does this sound familiar? It's very similar to import into the ER, which all of you guys may have noticed this weekend, right? Okay, so there are some differences though. So for example, you're not co-translationally imported into the mitochondria. It's post-translational. So proteins are post-translationally imported into the mitochondria. And there's a signal sequence, but it's not a specific sequence. It's a structural sequence. So it's a sequence that provides for the ability to fold into a helix with positively charged amino acids on one side and hydrophobic amino acids on the other side. Okay, so it would look like this, where all your reds would be on one face of your helix. Okay, because remember your amino acids, those properties are what allow for interactions. And so this structure is recognized by an import receptor. And the signal sequence is cleaved by signal peptidase, just like in the ER. Any questions about this? So it's just this sequence at the amino terminus that folds into this helix. So it's not like KDEL, or like it's not this specific correlations in arginine. It's a sequence that just forms this helix, which has this hydrophobic face to it. Okay? And so that's how it looks. This is the actual structure of a signal sequence from alcohol dehydrogenase that's bound to an import receptor. And so you can see it's looking straight down the center of the helix, and you can see the side chains sticking out. And here's your import receptor. And see there's this binding pocket, and that helix fits right in it. Okay? So as long as the protein has that conformation, then it will bind to the import receptor. Yes?
Is it just a signal sequence that makes
Just the signal sequence. This is just about recognition by the import receptor for import into the mitochondria. Oh yeah, I mean proteins are huge, so this is just, 15 amino acids or something. Yeah. Okay. Protein translocation occurs through translocons, just like ER, except for there are more options and there are two membranes to deal with. Right, so there's the Tom complex. I love Tim and Tom. That's a translocase of the outer membrane, Tom. And then this is the translocase of the inner membrane, Tim. So there's a Tom and then there's a Sam. And then there's a OXA. So these are just ways that these proteins get into the mitochondria. Okay, so the major steps of import. This is just the general picture. So you've got your protein outside the mitochondria, and it has a signal sequence, and that signal sequence is going to bind to the receptor. That's what I just showed you with that helix binding to that big gray blob, right? And then it will be inserted into the membrane. So the Tom complex, that translocon, actually inserts it through the membrane of the mitochondria, and it goes all the way into the matrix, which is weird, right? So your question might be, wait a minute, how's a folded protein going to get into the mitochondria? So actually in the cytosol where it's translated, it's bound by chaperones. And the chaperones keep the protein from folding and aggregating. Okay? And that way it's mostly unfolded and able to get through the translocon. So the unfolded protein peptide is fed through the translocon, and then it goes into the matrix, and then the signal sequence is cleaved in the matrix, and then you've got, so this is for matrix proteins that are going to the matrix of the mitochondria. And then you've got your mature protein that's folded in the mitochondria. Okay? What do we do? 2 minutes left. ATP hydrolysis and the pH gradient drive transport across the mitochondrial membrane. So you need ATP hydrolysis in the binding and the unbinding of HSP70. So remember that's bound, that's the chaperone, that's bound to the protein. That's bound to the receptor in the signal sequence. And then you have this electrochemical gradient. Where is that coming from? Why is there a gradient across the inner membrane of mitochondria?
Or oxidative phosphorylation.
Yeah, electron transport, right. And so you've got that gradient normally, right? Just from that. So that's also being used in import of proteins into the matrix. And then you also have and so then you also have ATP in the use of the mitochondrial HSP70 in import of protein further into the mitochondria. And then this is just showing that the proteins can also be membrane proteins, or proteins that are in the membrane of the mitochondria, right? And so they'll also have signal sequences, and then they'll have stop transfer sequences, just like ER. So you'll get translation all the way in through both translocons. You'll get cleavage of your signal sequence, and then when you hit your stop transfer sequence, it'll be moved into the membrane, which is similar to the ER. And then the other thing is sometimes you will have you'll have your signal sequence cleaved, and then you'll be on the inside. So you might be just go. I'm like, it's time. Okay, we're gonna still cover this, and maybe there'll be one magical question on the answer exam about it.